Thank you very much. And my name is Al-Masha RH. I'm from the atherosclerosis research unit at Al-Masha University, and this is our group logo. As you see, we have pigs and mice and atherosclerosis process all over. I'd like to thank the organizers for letting me present our model. And the entire motivation background behind the animal model is to produce pigs, in particular mini pigs, the Yucatan Minipig, who can function as a model of human-like atherosclerosis for the purpose of research in the field. And just a bit of background about this particular mini pig that was cloned. All the cloning procedure and all the effort that went to that is primarily done by my supervisor, Associate Professor Jacob Benson at Al-Masha University. I was not involved in the cloning procedure. It was a somatic cell nucleon transfer, and as I understand, the first large animal model that was done in with that technique. And what was inserted in these pigs was this protein, just called PCSK9 for short. It's a protein that is it's human isoform that was inserted and binds to the LDL receptor on the surface of hepatocytes, thus inactivating the LDL receptor. And so the pigs become functionally LDL receptor deficient, which leads to rise in plasma LDL cholesterol, and and I'll show the data, that leads to high cholesterol and atherosclerosis. From the original research that was done by Jacob Benson, we have obtained two founders. This was the first generation cloned pigs, two founders who were transgenic, and they were mated with a lot of wild type Yucatan sows, and this was the result of the crossing. As you see here, there were two insertions of this protein on chromosome 9 and chromosome X. So naturally, the offspring, we could get some wild type males, we could get males with chromosome 9 insertion, the distribution is approximately 50-50, and the females, we had some with chromosome X insertion and some with both chromosome 9 and chromosome X insertion. And of course, this breeding did not result in any wild type females, because all of them inherited the chromosome X. And the main hypothesis was to test that the transgene led, in fact, to increase LDL, led, in fact, to increase plasma cholesterol, led, in fact, to atherosclerosis compared to the wild type pigs. And we also tested the difference between the sexes, so females versus males also. This is the data from the pigs before they were put on a high-fat, high-cholesterol diet. So this is on normal diet. And what we see is that while the wild-type pigs had cholesterol levels as expected for a pig, about 2 millimolars, this is total plasma cholesterol, we can see that the transgenic males, who had only one insertion on chromosome 9, they had higher, significantly different higher cholesterol. The females a bit higher, and the females with the X insertion and with the X9 insertion still higher. No differences between these three groups, but all were significantly different from the wild type males. And on the right here, we see size exclusion chromatography. This is basically the cholesterol fractions separated by size. And what we see here is that there is a little bit of VLDL, a large top increase in LDL, and then also some HDL. And these are pooled data from the wild type group. This is the white circles and the black circles. This is the transgenic group. And as we see, there is an increase primarily in LDL in the transgenic males compared to the wild type males, and the other fractions, VLDL and HDL, are essentially the same. So this tells us that the theory that LDL receptor is inactivated and LDL increases seems to be correct. We also did look at the the hepatic LDL receptor levels. So this is a Western blot indicating how much LDL receptor was in fact to be found on the surface of the hepatocytes. And this is the mature LDL. And while there's a lot of LDL receptors on the surface of the hepatocyte, in the wild type pigs, we can see that the levels are diminished. This is the quantification of the plots here, and you see that the wild type pigs have maintained the level of LDL receptors, while the transgenic pigs are essentially LDL receptor deficient on the surface of their hepatocytes. This is the same experiment done with a blocking agent for the LDL receptor. You see all the bands disappear here. We also looked at, do you hear me? Do you know this one? We also looked at the LDL clearance. So we injected purified human LDL into the pigs, and we took sequential blood tests to see how the pigs were at clearing this human LDL. So this is ours from the point of injection, and this is human. As you know, ApoB is a protein on the surface of the LDL particles. So by measuring human ApoB, we could find these injected LDL particles in the pig blood. And what we see here is that, again, the white circles are the wild type pigs, and they were the LDL clearance capacity was maintained in the wild type pigs, while it was diminished or at least retarded in the transgenic pigs. And there was, of course, a significant difference between these two clearance plots. So what we have established here is that there's less LDL receptor on the surface of the hepatocyte as expected. And we also established that the clearance is retarded in the transgenic pigs compared to the wild type pigs. Now, we did go further to look at the phenotypic initiation of atherosclerosis. And for that purpose, we put the pigs, put them through a one-year trial where they were put on a high-fat, high-cholesterol diet. The high fat is 20% lard, which is essentially melted pig, and high cholesterol 2%, supplemented to the normal pig chow. So the rest of the graphs I'll show you will all be concerned concerned about the development in different plasma proteins and markers after the pigs were put on a high-fat, high-cholesterol diet. So this is weeks past the start of the high-fat, high-cholesterol diet, which is marked as time point 0. This particular plot is over the plasma PCSK9. Remember, this is the protein that leads to all the misery about increasing LDL. The wild-type pigs do not express this human form of the PCSK9. Of course, this is expected. Looking at the other groups, we have the, this is the, okay, so we have, let me look at the legend. This is the transgenic females, this one, the transgenic males are the circles, and the females expressing 2 chromosomes are the upward triangles. And we can see that all pigs did express increased levels of PCSK9. For some reason that we cannot explain, these 2 groups exhibited increasing levels of PCSK9 after they were put on high-fat, high-cholesterol diet, but that plateaued about 24 weeks or 30 weeks into the experiment. And we see here that there is, in fact, a difference between these 2 groups and the transgenic female group expressing only one transgene on the chromosome X. Very well, so this increase in plasma PCSK9 led to similar increase in cholesterol in all groups. So even though there was difference between PCSK9 expression in plasma between the different groups, the transgenic groups, there was a similar increase in cholesterol in all groups compared to the wild type group. Even the wild type group increased in plasma cholesterol when it was put on a high-fat, high-cholesterol diet. This was expected. But what we see is that the increase was more than 2-fold in the transgenic pigs being LDL receptor deficient. And looking at LDL cholesterol, of course this was total cholesterol. Looking at LDL cholesterol, we can see that the increase in total cholesterol is explained, in fact, as expected, by an increase in LDL cholesterol. Looking at the other cholesterol fragments, we can see that HDL cholesterol, there's no difference between the transgenic group. There's an initial increase in all groups, including the wild-type group, when put on the high-fat, high-cholesterol diet. The reason for this we have not investigated. But interestingly, we found a significant difference between the transgenic groups and the wild-type groups. So the transgenic groups ended actually having less HDL cholesterol compared to the wild-type group. We don't know the reason for this either. Triglycerides, we can see that at the start of the experiments, the pigs exhibited fairly high levels of triglycerides, all the groups, and there was a decrease in triglycerides. So these pigs were put on a high-fat, high-cholesterol diet, 20% fat, but in spite of that, they had a decrease in plasma triglycerides. This has been described previously in other pig models, and the reason of it also is unclear. But the triglyceride levels were fairly constant throughout the study, the remainder of the study, and the transgenic pigs did exhibit increased levels of triglycerides compared to the wild-type pig. So this is also another mechanism that we cannot account for. All right, so this was the plasma cholesterol levels and cholesterol fractions. Now what about atherosclerosis? Do they in fact encounter atherosclerosis? This was the whole point by introducing this pig. Now what we see here, I'll start by introducing the picture here. This is taken from one pig. This is the aorta split up in three halves, three parts. The thoracic aorta, so this, the heart will be somewhere here. This is the ascending and aortic arch. This is the remainder of the thoracic aorta. This is the abdominal aorta to the level of the kidney arteries, and then the rest of it to the level of the bifurcation, or rather the trifurcation in pigs, because there are three arteries, including the tail artery. All that is colored red is colored by Sudan 4, which a stain that is specific toward atherosclerotic regions. And what we see here is that there's plenty of coloring in the thoracic aorta. The picture quality is a bit inconvenient in the abdominal aorta, but we see some reddish coloring, and also we see raised lesions. All what you see here, that and this and that and that, these are raised lesions. So we have flat lesions at the thoracic aorta, raised lesions in the abdominal aorta, quite as expected and described in man. And this is the quantification. Wild-type pigs are just below 20% of total aortic area. Transgenic males, this is the main comparison, transgenic males, more than twofold. That's significant difference, no significant difference between the sexes. And going further to look at some of the other vessels that we also investigated, we're still at the aorta. These are the raised lesions. They depict the same tendency, significant difference between transgenic males and wild-type males, no difference between the sexes. The iliofemoral artery, the same thing, difference between transgenic males, wild-type males, no significant difference between the sexes. The coronary arteries, unfortunately, although we might see a tendency toward increase in the transgenic males compared to the wild-type males, but that was not significant. But please keep in mind that we have few observations here, so if we could have had more pigs, we might have achieved significance in this area too. But the interesting thing is that if we look at, this was plaque area and plaque volume, but if you look at plaque staging, according to the staging of Vermani and co-workers, where you can see we have low stage, normal or intimal thickening, xanthoma, pathological intimal thickening with extracellular fat deposition and finally fibroatheromas. There's significant difference between transgenic males and wild-type males and all the vessels, including the coronaries. So even though we didn't have more volume, significant volume, we did have more advanced atherosclerosis with transgenic males compared to the wild-type males. I will conclude by showing you a couple of examples to why pigs are more interesting in modeling human atherosclerosis compared to mice. So many of the features that we see here we don't see in mice. This is a a plaque from a pig exhibiting intimal thickening. This is it in greater magnification. This is the intima, is clearly thickened compared to what we see in normal pigs. This is the next stage. This is a pathological intima thickening with a lot of foam cells that we see here, a lot of foam cell deposition, and also traces of extracellular fat. So this is a pathological thickening. This is it in smaller magnification. And finally, we have this is the nice image that we look for when we do atherosclerosis research. This is the fibroatheroma. It exhibits a lot of the qualities that you have in advanced human atherosclerotic lesions. You have a fibrous cap, a thick fibrous cap. Now, all this is atherosclerosis. All this is encroaching on the lumen. The lumen seems to be narrow. The media is just a thin shell around it all. You have calcium, a lot of calcium deposition, and necrotic core, fibrous cap, new vascularization. These are new vessels, and also traces of extracellular bleeding into the plaque.
Can we come to the end?
Yeah, we're done now. All right, so this is the conclusion. Yucatan Minipigs, novel model of familial hypercholesterolemia and accelerated atherosclerosis. I would like to thank the collaborators, especially my supervisor, Jacob Benson, who conceived the study and all the work on the cloning, etc. All the rest of the group who worked very hard on the project, and all the collaborators and fundings for the project. Thank you very much.